For this mini lecture in Module 9, we're going to be looking at factorial design ANOVA. So far, you've learned about a one-way ANOVA, where we have multiple groups, but it's only one factor. So this is the same as a one-way ANOVA or one-factor ANOVA. You're going to see both of these. However, more complex designs are often necessary, where we might have two or more factors that we want to look at, and this is called a factorial ANOVA. So for instance, we might want to ask, do women live longer than men, and does being right-handed or being left-handed matter? In this case, there's two factors that we're looking at. We're looking at handedness, so being left or right-handed, and we're looking at sex, so men versus women. And our dependent variable, or what we're measuring, is lifespan. A one-way ANOVA would be just simply asking the question if women live longer than men. However, in that case, we'd only be comparing two things, so we could actually just do a two-sample t-test or a one-way ANOVA. However, the fact that we're interested in handedness and sex makes this a two-factor ANOVA. So we have our two different factors and our dependent variable, and that there could be some effect of sex, so perhaps females live longer than males. There could be some effect of handedness, so maybe right-handed people live longer because the world is made for right-handed people, unfortunately, if you're left-handed. And there could be more, I don't know, scissor accidents or something associated with left-handed people. And there could be some interaction. So that means that there would be a differential effect of handedness and sex on lifespan. So being left-handed and female might have a different effect than if you were left-handed and male, for instance. Or if there's not an interaction, that means that there would be a consistent effect of handedness and sex on lifespan. We're going to go through these different scenarios and look at some graphs to explain what we might see. Assume that we have data for independent individuals on what sex they are, what handedness they have, and their lifespan. Now, one important thing to note is that there's nothing repeated measures about this. Each individual we just have one set of data for. And also in designing a study like this, we would probably want to include unrelated individuals to make sure that each individual that's included in our data is independent. Here we have some data. Factor A is handedness, and that's on our x-axis, and you can see left and right-handed, indicated by L and R. We have women in red, men in blue, and our dependent variable is their lifespan. So each circle is the mean. And then we're just connecting these with lines so that we can see the patterns. If our data looked like this, what would we conclude? Well, we can see that the line for women is consistently higher than the line for men, so we would conclude that women would live longer. We can also see that the points for right-handed individuals is higher than the points for left-handed individuals, so we might then conclude that right-handed individuals live longer. And because the two lines are parallel to each other, there's no interaction. So in this case, we would say that right-handed people live longer than left-handed people, and that women live longer than men, and there's no interaction. Of course, the statistical significance we would look at a table. In this scenario, we have a significant interaction because the lines cross. So let's talk about what we see here. In this case, if we looked on average at the effect of handedness on longevity for men and women, so for now we're not caring if it's men or women, we're just looking at handedness, so the main effect of factor A, well, the averages are probably pretty similar in the lifespan of individuals who are left-handed or right-handed, so there's likely not a significant effect. When we look at men and women, regardless of what handedness they have, well, they would average out to be about the same as well. However, because the lines actually cross, we would have an interaction occurring here. So in this case, we would say that left-handed women survive longer than left-handed men. However, right-handed men survive longer than right-handed women. So because the lines cross, we have an interaction. Whether or not that's statistically significant, we would have to do the analysis and look at our F statistic. So who lives longer? Well, it depends on both the handedness and their sex. So we have some interaction between these two different factors. So when we look at statistical hypotheses, we're going to have one for each main factor and then one for the interaction. So for factor A, which is handedness here, our null hypothesis is that the means are the same. Our alternative hypothesis is that at least two of the means differ, right? This is an ANOVA analysis, so we could have multiple groups. These are general hypotheses here. For factor B, which in this case is sex, our null hypothesis is that the means are the same. Our alternative hypothesis is that at least two of the means differ. In the interaction, the null hypothesis is saying that there's a consistent effect of the factors on our dependent variable, and our alternative hypothesis is that there is not a consistent effect. When we look at a factorial ANOVA, again, because it's an ANOVA design, we're still going to be partitioning our variation. So recall for a standard ANOVA, we have our total sums of squares. Some of that is due to a treatment effect, and some of that is due to just random sampling error. When we have a factorial ANOVA, some of that variation is going to be due to the influence of factor A and the different levels associated with that. Some is going to be affected by factor B. We're going to have an interaction, and then we have error. So we're partitioning it into more pieces. This is what an ANOVA table will look like for a factorial ANOVA. So we have our source of variation on the left-hand side, then our sum of squares, degrees of freedom, mean square, our F statistic, and our p-value. You'll see with the different rows, so the top one will be factor A, the second one will be factor B, the third one is the interaction, the fourth one is the error, and then at last we have the total. So if we look just at handedness, of course we have our sum of squares and our degrees of freedom. Our mean squares are our sum of squares divided by our degrees of freedom, and then we have our F statistic, which is calculated by, here we have 100 divided by 5, so that's handedness divided by the error is going to equal 100 divided by 5, which is 20. For sex, it's going to be 78 divided by 5, which is 15.6. Our interaction is 67 divided by 5, which is 13.4. So when we're calculating our F statistic, it's whatever factor we're looking at, or the interaction, divided by the mean square error. And then we have our p-values here as well. So given this scenario where we have handedness being significant, sex being significant, and the interaction being significant, what would that look like on a graph? So when all of the effects are significant plus some interaction, if you have data that looks like this, we would conclude that women live longer than men on average, so that is regardless of handedness. Women are living longer than men if we average across both handedness of women and both handedness of men. If we look just as handedness, individuals who are left-handed live shorter lifespans than individuals that are right-handed. So now we're going to average the men and women together and only look at handedness. And then we see that there's some interaction. So in this case, left-handed women live shorter lifespans than left-handed men, but right-handed women live longer than right-handed men. So this is what you would see if all of the effects were significant and the interaction. So in this example, each of the factors that we looked at had two levels. So factor A, handedness, is either left or right-handed, right? So there's two levels. Factor B, which is sex, so here we have men and women, and that's two levels. But there are going to be more complicated examples as well. And so we're just going to walk through some figures that I've pulled from a different textbook that I actually quite like and sort of drive this home when there are more levels for each of the factors. So this is some data. This is actually from an ecology textbook that I've used to teach biostatistics before, but I just like the graphs. So the details don't really matter. At the end of this, I'll ask you to think about some more health examples. So in this case, on our x-axis, we have factor A, which is some predation treatment. So maybe predators are present in different amounts or absent, and there's 4 different levels here for the predation treatment. Factor B is the substrate, so if it's cement, slate, or granite, and that's representing the different lines. So the substrate has 3 different levels. And our response variable is the number of barnacles that are recruiting. So barnacles live on rocks and coastlines of the ocean. The bars here represent standard deviations. You can just ignore these. We'll just look at the lines and how they look relative to one another. So in this first graph, there's no significant effect of anything. So NS means not significant, right? When you look across predation treatments, they all seem really similar, right? Those mean levels. When you look at substrates, well, all the lines overlap, so they're not significantly different from one another. And there's no significant interaction because all those lines are really tightly packed together. In this scenario, if it looked like this, again for factor A, so if we look across the different predation treatments, well, the third one looks really low relative to the other ones. So this data here is suggesting that it's statistically significant, that one of them is different from the other ones, and this one's quite a bit lower than the others, so that might be where that's coming from. Again, for substrate, all those lines are really close together in parallel, so there's nothing significant going on there. And then again, because those lines are basically overlapping, there's no significant interaction. For C, the one on the upper left, when we look at the predation treatment, if we just ignore which substrate it is, if we just look across the predation treatments, on average they look quite similar, so that would not be significant. But now for substrate, the lines are separated from one another according to, granite is at the top, slate's always in the middle, and cement's at the bottom, so there's a significant effect. And then there's no interaction because those lines aren't crossing. They're all parallel. The one on the lower right, now we see that when we look across the predation treatments, that there is variation, so it's still dipping down, for instance, at that third treatment. So predation is significant. The lines are parallel but spaced apart for substrates, so there's a significant effect there. But they're not crossing, the lines aren't crossing, so there's no significant interaction. When we look at this one, it suggests there's no significant effect of predation. So if you look at the 4 different treatments, and if you didn't care what substrate there was, you just took an average, that means that there's no significant difference across the different predation treatments. When you average across the predation treatments, when you're just looking at the different substrates, there's also no significant difference, but the lines are crossing, and so there is an interaction. And then finally, the other scenario that you could have is the predation treatment here is not significant, because if you forget about which substrate it is and you just averaged all those together, they'll average out to not be different across the treatments. Because, for instance, on the far right-hand side, although the blue line goes lower at the bottom, well, the black line's highest at the top, so those will sort of cancel each other out and end up not being different overall. For the substrate, the lines are separated from one another, so there is a significant effect, and there's also a significant interaction. In this case, they are not crossing, but the blue line dips down way at the end, whereas the black line goes up. So when you only have a 2x2 design, right, when we look at handedness in males and females, often you'll see it crossing, but in this case they're going opposite directions, so you could also see that happening as well. So these are different scenarios that you might see. Some other examples that you can think about, and you could draw these out for yourself and try to see what the main effects and the interaction would be. For instance, would be to look at impacts of maternal age. So here are 3 different age classes, and smoking status, so yes or no, on the birth weight of children. So that birth weight would be the response variable, and the first factor would be maternal age with 3 levels. The second factor would be smoking status with 2 levels. Or you could look at something, for instance, the impacts of alcohol consumption. So that's our first factor with 3 different levels, and diabetic status, again with 3 different levels on the response variable of blood pressure. So given these scenarios, you could draw out some diagrams and think about what they would look like if there was one of the main effects had an influence, if the other one did, and if there was or was not a significant interaction.